This video is about glycolysis and pyruvate oxidation. Here's the overall process. You take a 6-carbon glucose, and overall you break it into 2 molecules of pyruvic acid. The main ideas here is that you're producing a small amount of ATP, and NAD+ is picking up hydrogen and electrons. This will later go to the electron transport chain and result in a lot of ATP production. During glycolysis and then later in the citric acid cycle, we have something called substrate level phosphorylation. Whereas the substrate has an inorganic phosphate, the enzyme cradles the substrate phosphate complex with the adenosine diphosphate and helps this process happen where adenosine diphosphate links to the third phosphate, making ATP. Here's the overall process. In the first part of glycolysis, you have a 6-carbon molecule. You do a little bit of energy investment with 2 ATP. It rearranges to fructose diphosphate, and you can see the phosphates here and here in G3P. In the second phase of glycolysis, you have 2 things happening. One, NAD picks up some electrons, which it takes to the electron transport chain. And the other one is that 4 ATP are made, ending in 3-carbon pyruvate, which goes over to pyruvate oxidation. This is just a different picture of the same thing. Here's the whole thing that we are looking at in 4.2, glycolysis, pyruvate oxidation, and the smaller molecules goes through the citric acid cycle, and finally everything flows into oxidative phosphorylation at the electron transport chain. We have a 6-carbon molecule here. We have the energy investment phase here with 2 ATP, and then finally we have the energy payoff phase where 4 ATP are produced and 2 NADH are produced, which goes to the electron transport chain. Pyruvate goes to the pyruvate oxidation in the next phase. Here's the main biochemical reactions. Here we have glucose, 1, 2, 3, 4, 5, 6 carbons. Hexokinase works on it. It is phosphorylated, as you can see here. So we have glucose going to glucose 6-phosphate. So 1, 2, 3, 4, 5, 6. Phosphate is on number 6. In this case, we have glucose 6-phosphate going to fructose 6-phosphate. If we look here at glucose, we have a hexagon with 1 carbon above the chain. Here we have fructose, which is a pentagon with 2 carbons above the ring. So it's just a isomerization, a rearrangement. Over here, we have fructose 6-phosphate going to fructose 1,6-biphosphate. So ATP is going to ADP, and another phosphate has been dropped off on the number 1 carbon. This is considered a phosphorylation reaction. So this is a major split that's happened. So we have a 6-carbon molecule, and it's broken into 2 3-carbon molecules. We have what we'll call G3P. We have 1, 2, 3 carbons with a phosphate. This other molecule, let's call it dihap, will also be converted to G3P. So we're halfway through this series of reactions. So what just happened? This is the energy investment phase where we have glucose. 2 phosphates have been added, and we've broken the molecule roughly in half. Over here we have G3P and NAD+, which is electron carrier, comes and picks up some electrons, and hydrogens are also taken and put into the cytosol. Inorganic phosphate is added, so we go from 1 phosphate to 2 phosphates, as we can see from the name, 1,3-biphosphoglycerate. So over here we have two phosphates, and we have ADP coming in and taking one away, so now we only have one. This is by substrate level phosphorylation. Over here we have a simple rearrangement. So this phosphate on number 3 has been now moved to number 2. Over here, there's a loss of water. So we can see that OH and H has been removed from this molecule. Now we're at something called PEP. In this last picture, we have PEP and ADP is picking up one of the phosphates, and now it's not in the final product, which is pyruvate. So we have a 6-carbon glucose breaking into 2 3-carbon molecules of the phosphate, finally ending with a 3-carbon molecule named pyruvate. So now we're able to go into the next phase, which is pyruvate oxidation. So we have pyruvate. It goes with a transport protein into the matrix of the mitochondria. And 3 things are happening. 1, carbon is removed. So we go from a 3-carbon molecule to a 2-carbon molecule. NAD plus is picking up electrons, plus we're having hydrogens being picked up. And coenzyme A is attaching itself to acetyl-CoA. So now it's ready to go into the next phase. So we have a decarboxylation happening, a dehydration, and a reaction with coenzyme A. Now acetyl-CoA is ready to enter the citric acid cycle. So the final result is a net yield of 2 ATP, 2 NADH, and in pyruvate oxidation, we have 1 NADH being produced. And NAD is used for shuttling electrons. The purpose of which is to take the electrons to the electron transport chain for oxidative phosphorylation and ultimately to make ATP.